Yeah.
Hi,
How are you?
fine.
Good. Yeah, glad you could make it. We had a little trouble with the Jitsi space, but we can move into this space and it's back up.
Yeah, Seven. I think it's better than the Jitsi.
Yeah.
It's more stable right now.
Yeah. Let me wait for a couple more people. I think Anson will be back. And I don't know who else. So, welcome to the meeting. This is the last couple days before the beginning of Neuromatch. So, everyone's excited. They had a party last night because there were a bunch of people who were preparing for Neuromatch, and there were a lot of things going on, a lot of things being prepared. I helped with some of the videos, and so that's going to be. And they had a sort of a little party last night on Crowdcast. So I think I put a link in the Slack channel if you want to check that out. So going into Neuromatch, I guess we're going to still have our meetings. Next week we won't have a meeting on Saturday because I'm going to be presenting at a conference at that time, so I can't be here. So we'll move it to some other time. But we'll have the meetings throughout the Neuromatch period. We'll also have groups in Neuromatch. So the project groups, we're going to have a project hosted by this group. Let's see if I can find it. Well, I can send out the detail. I think I sent out the details in the Neuromatch channel, but it's called Feedback of Complexity Loops. I'm not sure how the projects are going to be handled, but if you want to join that project, you're welcome to it. I think it's number 8, but I don't know how they're actually going to give people the option to join groups. So.
Yeah, I was curious about that because I know the pot. I think it would be good for all of us to give our own Neuromatch updates, maybe a little bit. And we don't do that. I don't know if we want to do that now or whatever. But I had quite a time getting into my pod because I wasn't in the system for a very long time, apparently, and then there was some intake issue. But it's been resolved, and I think it's Patrick Minot, one of the main tech guys. He was really helpful, and first-time conferences of this magnitude. I don't have any. I have zero complaints about Neuromatch Academy because it's really literally unprecedented as far as I can understand. But I have no idea about the projects. I finally got them into my pod space, and I'm familiarized myself pretty much with the NeuroStars things, and some of the content that I've had access to. But I'm curious about other people's experiences, or even in the Neuromatch Slack, I haven't really seen anything about the projects. I might look now, but I don't know. But I'm very glad. I mean, the one that was mentioned here looks pretty cool, so that's good.
Yeah.
I'm trying to see where Anson is at.
Yeah. Did Sebin or Bobby? Oh, here he is. Did Sebin or Bobby want to comment on the Neuromatch projects?
I haven't actually heard anything about them either. I've been kind of trying. I've been in a similar boat to Jesse, but kind of the opposite. I got added to both the Observer and the Interactive track. So I've been trying to handle that for the last few days. But that is all squared away. I'm gonna go ahead and hop on NeuroStars and look through the forum today and see what's up. But
yeah. Sebin?
Yeah, actually I am on the Observer track because I was just worried that I will be able to keep up with the requirements of the other track. So I was just going through the Python tutorials, and as just I said, it's really incredible because this is really unprecedented, and it's the level of organization and the way it can be turned to be useful is very much appreciable. It's really awesome.
Yeah, good. Yeah.
And I'm a bit worried about the project thing because I am because whether it will be too tough for newcomers like that. Okay.
Yeah, I'm not sure how they're going to handle. I mean, like obviously there'll be groups, so there'll be like you'll have people with more experience and less experience in the group. So I don't know how they'll do it, but it's going to be like you're going to have a mentor, and then the mentor is going to help you through the project. They're going to be, I think, like they'll have data sets available for people to use, but I don't know, like beyond that, I don't know what the details are.
It sounds like the project mentor will be separate from the person leading your pod. Or is that not the case? I don't really know.
No, I think you can pick your own project. I think it's independent of that. So, but I think it's just something that you can do. Like you can pick a topic and apply what you're learning to that project, but it's independent of the pod's system. So,
okay.
Yeah. So, let me see if I can share my screen and show you this one here. This is for the benefit of people who couldn't make the meeting. So Sunidhi couldn't make it today. Her grandfather had an issue and it's a family emergency, so she's not going to be here. She was going to do some presentation on some of the things she found about aging and neurodegeneration and that. But I actually have a talk on that. So that's So she'll be maybe catching up with us later.
My camera's working here.
So, can everyone see my screen here?
Yeah.
Okay. So this is the Neuromatch Academy, our repository, and this is the epistemological directory that Jesse helped set up. And one of the things I've included in here is complexity of feedback loops. So if you go into this area, this is this project idea for complexity of feedback loops. And so this is one of the projects. I don't know why it's not showing the final version here. But yeah, I think it's this one. Sometimes when you commit things in GitHub, it doesn't come out right. I mean, it comes out like this. I don't know why. Anyways, well, this has a description of the project in it, and I don't know why it's not displaying the way it's supposed to, but I guess view file. Okay, there we go. So this is the project idea. It's pretty theoretical, and there's another version of this that focuses mainly on vision. I didn't include that. I don't know if I included that in this, but if you'll see when you select projects that there's a version that has that's deals with visual pathways, and then this version, and so there's a specific question. So this is something that we've been exploring in this group. It's about different types of representational models. So the idea is that if you have like a no representation versus a significant type of representation where you have symbolism and things like that, and we can talk about what those mean. But basically, the idea is how do you model feedback or behavioral feedback, maybe more specifically, with different degrees of complexity. And so if you want to model feedback, you can model it using data, you can model it using a neural network. But how do you represent feedback and how do you model it? And so I was trying to make this clearer for people to understand. But this is something that, if you're interested in, we can, kind of think about it in terms of like a specific data set or a specific system, and it'll become a lot clearer. So this is it's in this directory, so complexity of feedback loops. If you're interested in that project, then when you make your project selections, we'll be in touch, of course, and we'll work something out. So and I think you start the first week on the project selections. So, any questions about that or any comments? Jesse, you're muted. I think you're trying to keep talking.
Yeah.
It's my camera for and my mic muted. I updated the file to a README file, so it will show in the folder. But I'm interested in the project in general. I'm curious about data sets. I know that this is the same one that we were talking about in the chat, right?
Yeah.
Or where it was like, and there's someone using an eye visual data set.
Yes.
As well. So I'm very curious to use that. I don't know. Is it such a thing where the projects need a certain number of people to do it to get selected, or is it just? I think it's a mechanic of the Neuromatch that I don't know anything about yet. It's the project stuff.
Oh, they've already gone through the selection process. They've just given us they gave us some themes. So we submitted.
You can just pick anyone.
Yeah. So they're going to give you a list of things to choose, and then that should be on the list. It may not. I don't know if they made a final cut, but they had the themes that we proposed.
Okay.
And I don't know. You might, they may even have people proposing their own, but I think this is a pretty for this group. It's matching at least some of your interests, and then I think it would be good if we could just get a little bit of data and work it out a little bit more. I don't think you have to finish it I don't think you can do a finishing. You can finish it up in 3 weeks. I think you just have to basically get into it, because you're already doing a lot during the week. So it's but I think just to get a good start on it, I think is usually the way they view it.
Yeah, it kind of has the shape of something that would be interesting to do. Even in it's something that I might want to work on. Excuse me. Even if it's not a project available in Neuromatch itself, I think it'd be good testing for a lot of things that I want to do otherwise. So, yeah. I'm interested in that.
Yeah. Anyone else?
I'll do what Jesse said. I feel the same way.
Okay.
Yeah, that's what I heard Jesse said.
Yep. So yeah. So the next order of business is we have our DevoNN abstract, extended abstract for artificial life. That was accepted, and then this week we made the video for the talk. So thank you to Jesse and Z Gong, who's not here, but he attended the version. We recorded the talk. I gave the talk, and then Z and Jesse gave comments. And then I found out that the video was corrupted in the file. So, so I recut the video recording. I had to show the slides because the slides aren't showing up right. And so I got that out and I sent it to them, and they received it with the final version of the paper. And then we have that on YouTube as well on our channel. Let me look over this real quick for people in case. I know there are a lot of people on this as co-authors who weren't at the session. So let's look at this over real quick.
Yeah, just for people who aren't in the lab, I assume, are we looking at the abstract or the slides?
Well, we'll look at both. So here's the folder. So why don't we go through the talk first? Look at that.
It's basically everybody who was in the lab last year. And you guys have kind of seen stuff on. I don't know if you've anybody here. Maybe Anson did, but I don't think Z's been in any meetings. But you guys see from left to right, it's Bradly, Stefan, Z, Ankit, Sam Felder, who was actually the 2018 summer code.
Yeah,
person.
summer code, yeah.
And then the weird guy on the right, standing next to a Norbert Wiener poster at the MIT Museum is me. So,
yeah. So yeah, everyone kind of contributed somewhat to this work. It's not
me. Good.
Yeah, it's not like it's kind of like if you've done some work on this area, you got into the author list. I wanted to present on a number of ideas. This is the workshop logo, and this is the conference logo. So this conference is this coming week, during the week, and then the workshop. We recorded this talk, and then it's going to be on YouTube, and then we're going to have a question and answer session next Saturday, which is during this meeting. But and then so it goes through this idea of metabrain models, which actually relates to the project because we talked about representation-free and representation-rich models. And this is kind of the idea behind this metabrain model idea. So representation-free models rely solely on the structure of the empirical world. So it just processes data. Bradenburg vehicles are an embodied version of this. But neural networks are also kind of the same way. It's just processing data. And it's giving some answer. Could be like moving wheels, or it could be classifying pictures or whatever. Then the representation-rich model, which is this represented by the contextual geometric structures that are in this talk. It's a model that's representation-rich, so it has maybe semantic content, or it's like some sort of thing that gives assigns meaning to input data. So this is usually these models are implemented separately. If you think about good old-fashioned AI, that was very representation rich, but it was very brittle. You could have a lot of representation, where if it played chess, you might give it every single move ever devised, and then attach a symbol to it and say, if you're in this situation, deploy this strategy. And so that's how it would, but those were very brittle models. The representation-free models are pretty flexible, but also brittle in the sense that they don't process. Oh, hi.
Sounds like Angela.
I don't see list. Angela?
Not so much Jitsi.
Oh, she's in Jitsi. Why don't you send her a link to here? Yeah, I think she's in Jitsi because she couldn't make it into the room unless I let her in. So.
Let me see. I thought I closed it, but I'll look.
Okay.
I'll
go back to this. So that's basically the idea of metabrain models. Then we go through Bradenburg vehicles, and we go through this DVB brains and connectivity matrix. So we had a couple of different versions of what we call developmental Bradenburg vehicles, which are these Bradenburg vehicles where the topology inside of the vehicle develops. And so here's an example where you have a couple of nodes between the sensors and effectors, and then you have additional nodes that grow over time, and you have this kernel, which is the representation-rich model in the middle. And then we had one summer project by Stefan, which was using a genetic algorithm to grow brains of different topologies and select them based on their fitness. We also had a multimodal model, which was where the vehicle would encounter two different sensory inputs, and it would learn the association between them. Let's see what else is interesting in this talk. We talked about CGS models, which is something that Anson and Jesse have been studying prior to Neuromatch, the Academy. And so this is, I'm not going to really give this talk again, but I just wanted to give an idea of so these are the representations. You represent things like colors, were shapes on a geometric space, on like it projected to the side of a geometric space, and then the inside of the geometric space is where the associations happen between the representations. So there's an intersection within the space of different of the shapes and the colors, and they get associated in different ways and overlap. And so then that's related back to what the Bradenberg vehicle is experiencing, and it relates to how it might move through the environment or exhibit complex behaviors. And so then we have some future directions, and then a plug for the meetings and everything else. So, hello, Angela. How are you?
Oh, I just realized I was muted. Yeah, I'm doing all right, I guess. How are you?
All right, I'm doing all right. I was just going over the something that we submitted to a conference, and so we're going to be presenting on that this week. And
I don't think it was Angela that joined the meeting and said, "Hey," a little bit ago. I think it was someone else, and I have no idea who it is. So I'm reaching out to other people.
Okay.
Could be, but yeah, mystery.
Yeah.
Mysterious.
Yeah. We're also talking about projects for Neuromatch, and so we're pointing to the project that the lab is sponsoring a project on feedback loops. And so if you are interested, I'll be sending out more information and then you'll find out. But there's a project called Complexity of Feedback Loops. And so the lab is doing one track of that project. And so if you're interested in joining us, it would be great. It's very theoretical, but we've talked about like how we would bring data to bear on the problem and everything. So basically, it's the idea that we would basically be investigating different feedback loops, possible mechanisms for like regulation in a data set. So if you like think about like how behavior is regulated, like I just showed you, or how maybe like an EEG signal is regulated in the brain. I mean, there's a lot of stuff going on with feedback in the brain, but it's very, it's one thing to talk about it. It's another thing to show through analysis that it exists. And so it's a tricky thing. And I think it's actually quite valuable to show that kind of regulation, to show that it exists. Or what's even more useful, I think, is to show sort of, how maybe like representation in like the brain or something like that helps us to understand sort of the feedback. So in other words, if you have like a system like a pendulum, which is not there's no representation there, but it has a behavior, what are the feedbacks there versus something like an animal that has a nervous system where it can maybe locomote and it can adaptively move, but there are a lot of feedback mechanisms. And something like C. elegans, which is a small nematode, the behaviors are actually, they're pretty simple, and they have a very small nervous system, but the regulation there, the feedback mechanisms are actually pretty complex. And even so, something even that small, you have a lot of intricate feedbacks and such. So, it's a little fuzzy right now. We can talk about it later as to how to actually get our hands around it. So like I said, Sunidhi had a family emergency. She can't join us today. She was going to talk about some of the stuff on aging that she found, and there were some data sets. And so those might actually be something we could use, but I don't think it might not be so suitable for this project. But she said she found some things, but she couldn't make the meeting. So, but I did create a presentation for aging, and so I'm going to present that now. So can everyone see my screen? Okay. So let me go into this presentation. So this is on aging. So this is sort of an, we talked last week, we talked about computational stuff, Bayesian stuff. This week we're going to talk a little bit about the brain and how it works in, like, it's just a chunk of neuroscience, aging and life history and things like that. So this picture here shows, it's pretty busy, this cover, but this shows you some of the things that we think about when we think about aging. So, we think about aging, we think about the brain, we think about like the shape of the brain or the morphology of the brain, and that degrades over time as you age. And there are a number of diseases related to that. But you also have a lot of molecular and cellular mechanisms. And those can be very hard to understand, but I'll talk a little bit about some of those. And we also have a nervous system that not only consists of neurons, but also of things like glia. And so when we look at aging, we look at not only neurons and their condition, but glia and their condition. And there's all sorts of activity within the cells in terms of genetics and molecular mechanisms that change with age, and so they contribute to this aging. And so someone's brain, when you go from a teenager to an older adult, you end up with changes to the morphology, but you also end up with a lot of changes to the biochemistry and the sort of this cellular anatomy as well. And so aging, when we think of aging, it's really equivalent to something called neurodegeneration. And the reason I make that connection is because, oops, let me let this person in. Bobby's in. I don't know if Bobby's coming in twice or what. But I like your logo there, your network logo.
Oh, sorry. I went onto my phone for a second, so I joined from my phone.
Oh, okay. I like your logo though. It's very nice.
Thanks. It's the correlations between mRNA channels in a homeostatic model.
Oh, good. Yeah. Very apropos here. Yeah. So let's go back into the presentation. So the reason I make this distinction is because neurodegeneration can happen at any age, really. Now, this is a picture of a healthy brain in a 25-year-old, and this is a picture of a aged brain in an 80-year-old. And you can see that there are a couple things. You lose volume in your hippocampus, which we'll talk about. These gyri, which are like kind of holes in, places in the brain where there are gaps, those increase in size, and so that's all indicative of a brain that's, sort of disintegrating, or the cells are changing their morphology. But if you know anything about like football players who played football for years, sometimes they suffer from brain damage that is neurodegenerative. So you can see this in people who are much younger than a 25-year-old, or sometimes if even in 25-year-olds if they've suffered a lot of trauma. And so aging is it doesn't necessarily correlate with your physical age or your number. It correlates with how much trauma, how much neurodegeneration is going on. And so aging leads to a loss of axons, neurons, and tissue volume in key structures. It's related to lifespan, but not exclusively. Stress and inflammation play a huge role. And cognitive atrophy is also important. So if you use your brain, you tend to retain some of your faculties longer than if you don't, if there's cognitive atrophy. And there's some debate about brain training, but that's something that's not, like, the science of that isn't settled, but you'll hear a lot about brain training, there's some truth to it, but it view with a little bit of skepticism when you run across it. So this is what the human data look like, and this graph on the left shows like performance on different tasks that they've given people from age 25 to age 81. And you'll see that a lot of these tasks, such as inductive reasoning and spatial orientation and perceptual speed, they all go downhill with age. And so there's a pretty steep decline from 25 to 81, right? But there are a couple faculties that you retain, maybe even improve upon when you age. And the two that are highlighted here are verbal ability, or numeric ability, and verbal ability. So actually, those two are, you actually gain verbal ability as you age a little bit. And your numeric ability usually stays stable, although there's a peak at about age 45. So this is the effect of aging. It's not uniform across the brain. There's some cognitive faculties that are retained over age, but a lot of your skills go downhill. And again, if you engage in a lot of intellectual activity, your curve might look different. You might lose things a lot slower, and your curve might look like this instead of almost like a straight decrease. And so you can see them broken out here. This is a t-score, which is just a statistical transformation of the data. This is chronological age. This is reasoning. This is spatial orientation. And you can see that they decline, but that they're not necessarily, it's not necessarily uniform for everyone. This is another example. Here are the lines. So performance is preserved over age for world knowledge. This is where performance declines for a host of other things. These are on different types of tests that they've given. So these are specific tests for these different parts of cognition. So working memory, there are a couple of different tests. Long-term memory, there are some specific tests you can give. And so they've done this with different tests, because you could say, well, one test might not measure, the aspects that are degenerating, and you could have a debate about that, but they all are basically consistent. And this comes from a paper on video game training. And so, a word on video game training. Video game training can actually enhance. People say video games might be bad for people, but there are also some good things that video games can do, and one of them is it can enhance people's spatial attention and spatial cognition. And so they've used actually these video game stimuli to, examine people's ability to adapt in those ways. And they found some interest. There's been some interesting work on it. If you're interested, I can send you some papers on it. What can we do to reverse the effects of aging? So aging can't be reversed. All we can do is increase the number of neuroprotective inputs. So it's like, the things that help you stay younger, or the things that help you to stop, keep you from declining, are the things we want. But it's difficult to know what works in what context, and there are very few universal results. So it just depends, and it's on a person-by-person basis. But we know that things like cognitive activity and training can help, like video games, physical exercise, crossword puzzles. I mean, there's some debate about whether those are really strong effects, but nutrition, antioxidants, omega-3 fatty acids, calorie restriction. You'll find all of those in the literature. And again, there's a lot of debate on what works and what doesn't in certain individuals. And then genetic background. So there are genetic variants, there are epigenetic effects that people, some people with certain genotypes have. Sometimes if you do things like improve your nutrition, your gene expression is primed for neuroprotection, and so those things feedback into one another. But again, it's hard to know for individuals. It's going to be different. So this is an example of something called senescence. If we drill down to the cellular level, what happens in aging is that the cells, once they differentiate, they go through they can go through these mitotic divisions. And eventually the cells get old and they die. And there's some cellular regeneration in the brain, but not a lot after a certain age, especially. And so these cells eventually get old and die. And in doing so, they exhibit a lot of inflammatory responses. And a lot of these are molecular. But the end result is that the cells become dysfunctional and they die off. And so this is a motor neuron here. This looks like a squid, but it's a motor neuron. And then you have all these different types of neurons. So just as an aside, look at the diversity of shapes we have for different neurons, like the oligodendrocyte, the astrocyte, the microglia. Those are actually supporting cells of the neuron. You have neurons that are stellate and neurons that they have different shapes. And basically, they don't all look like this. This is a motor neuron where you get a really long set of processes. And that's the one you typically see in a lot of the popular depictions of a neuron, but there are a lot of different types of neuron. And neural cell. So astrocytes are not neurons per se, but they're neural cells. And same with microglia. But there's a lot of diversity in shape. But there are a lot of things that can lead to this senescence. Stress-induced cell death. So if you experience a lot of stress, your cells can age. The senescence of cells like glia, which are supporting cells, like I said, this is a microglia, that can lead to the destruction of a system. So if this microglia starts to age, it can affect the myelin sheath on this axon, and that can, disrupt the function of this neuron. So it's, in that case, it's a whole system of things that can be affected. There are system-wide signals such as inflammation. So when you suffer from inflammation, it can affect your aging of your brain. Also molecular mechanisms like telomeric shortening. So in your genome, your chromosomes have things called telomeres on them. And when your cells divide, the telomeres shorten, and sometimes they grow back. But if they get to a certain length, it's like a clock, and it relates to aging. So if they get too short, this cell undergoes this senescence and dies. And so there are all these and you'll find in the literature, there's a lot of literature on what causes cellular aging. And so this is one of the things, or one of the senescence, and then there are a whole bunch of things under senescence. But we also have things that happen at the anatomical scale, and one of those is Alzheimer's disease. And of course, you've probably all heard of Alzheimer's disease. This is where you lose your memory and you suffer from hallucinations or things like that, and it gets progressively worse. So we don't really know. I mean, we know it's a hereditary thing. Maybe it's due to environment to some extent. I mean, there's a strong hereditary component, but we know the sort of the pathology of the thing. And so on the right, we have this brain that's suffering it goes from a healthy brain to a severe Alzheimer's brain. And the brain shrinks in size, as you can clearly see here. These gyri, which are the holes in between these sulci, which are the tissue that's folded up on the top of the brain, those get increase in size. And if you look at a cross section, you'll see that basically it desiccates, the brain. You get all these things like plaques, which I'll show in a minute, and tangles in some of the tissue, and so it's a mess. This is an example of things that we observe in the phenotype. So we observe these neurofibrillatory tangles, which are just proteins that aggregate in the tissue. You get the loss of neurons. You get amyloid plaques, which are plaques that occur in the tissue, and they disrupt neurotransmission. All these things are kind of contribute to this disease phenotype. And so this is something that can happen with aging, and it's triggered by aging. We don't really know. I think we know that there's some, it can be early onset or late onset, but there is a lot of it is a molecular phenomenon. But we know that this is associated with aging. The other thing that happens with aging that's been observed is that the hippocampus shrinks. And so the hippocampus and this mediotemporal lobe more generally, which is I think this is sort of just kind of like the hippocampus, but this is the mediotemporal lobe in this region of the brain. This is in the rodent and this is in the human. So we have this rodent brain with the hippocampus and the human brain with the hippocampus. And so this whole part of the brain is heavily involved in spatial and episodic memory consolidation. Now, improved memory function in hippocampus may lead to changes in volume. So they've observed, and this is a little controversial, that squirrels that cache a lot of nuts, or people who drive taxi cabs in large cities, can experience a enlargement of the volume of their hippocampus just by nature of like having a lot, having to do a lot of spatial computations. And so that's one way that the hippocampus can change. It can grow, but it can also shrink, and that happens as you age, and this can lead to memory loss. So as the hippocampus growing can lead to some enhanced memory capacity, its shrinkage can lead to memory loss. And the shrinkage is due to age. But again, if you do things like if you do a lot of spatial activities, spatially based activities, sometimes video games qualify in that category. There are other things you might be involved with, like driving an Uber or something like that, that sort of protect or preserve that function, then that can mitigate that loss somewhat. I'm giving a lot of papers down at the bottom, so if you want to look at the slides, you can get these references. So as we age, our hippocampus tends to get smaller. But as you can see from this graph, it doesn't happen at a uniform rate, and it doesn't happen suddenly at a given age. And as well, it doesn't happen in every individual equally. So you just have these individuals. Each of these lines is an individual. And you can see that like in some of these individuals, you have an increase in size at early ages, like in your 20s and 30s, maybe even into your 40s. You have a few people where it's declining. And then as you get out into your 60s and 70s and 80s, you still have people who have the size of their hippocampus is increasing, even up into almost to age 80 for some people. But you see more and more cases of where it's declining. And in this case, after age 80, it declines rapidly. So you can see that sort of variation, how it works. Now, genes and behavior. So when we talk about genetic effects on a complex phenotype, we have to distinguish between a couple of things. So one of those are genes of large effect and genes of small effect. So when someone says there's a gene that controls X, you should be skeptical of that. So to do this in a quantitative manner, we can look at quantitative trait locus analysis, which is a way to understand genes and their alleles and their effects on some phenotype, whether it be behavior or something in some structure in the brain. But you have to be careful, and we can see from meta-analysis that even with these rigorous methods like QTL, that there's a problem in that a lot of the genes that are candidate genes aren't necessarily don't have a very strong effect. So I'll give you two examples. The study where they look at historical candidate genes for schizophrenia. They did a meta-analysis and then an expert evaluation, which is where they ask experts about their opinion about this candidate gene, of 25 candidate genes for schizophrenia. And in doing this meta-analysis, they found that there was no consensus in any one gene being a gene of large effect. So all the candidate genes they had out there that people say this is associated with this disease, this is associated with the same disease, none of those genes actually had a very large effect. And they looked across studies, so they wanted to see if it was consistently associated. And they didn't really find that. Another meta-analysis is where they looked at 150 candidate genes associated with osteoporosis. So this is not the brain, but this is something that's, again, with candidate genes, where you say this candidate gene is responsible for osteoporosis and so forth. They did a statistical meta-analysis on 150 candidate loci, which are different variants of the gene of interest. And they found that there was no consistent effect found for any of the candidate loci. So that means that, when we find a candidate gene, we usually find an association, but those associations may not have a very large effect size. Now, that being said, there's a lot of value in looking at genes or candidate genes or things that are associated with a disease, because there is a possibility that it could be a gene of large effect. and that could have a big effect on the phenotype. But we don't really know that. That we're not at the stage, I think, in science to know that. But when we talk about genes too, as well, we're actually talking about two sets of interactions: gene-gene and gene-environment interactions. So gene-gene interactions are when two genes interact in a pathway, and those effects can be suppressive, additive, or multiplicative. So we call that epistasis sometimes. And in epistasis, one gene influences another gene. So you can have dominant and recessive alleles for like eye color or like some disease. And so you often see the notation of genotypes where you have like, I think the notation people usually use is A, B, A and B, different combinations of these two letters. And so that represents like something about the genotype, whether it's, homozygous or heterozygous. And so that's one example of epistasis where your genes are interacting and it's giving you this phenotype. And so when you say there's a candidate gene, it might be a dominant gene, it might be a recessive gene. And so that makes a difference in how the phenotype is expressed. Another thing is gene-environment interactions. And this is maybe what we're most familiar with, where genes interact with the environment. So a single genotype can respond to the environment in different ways. So like a good example of that are genetic risk factors, where if I carry a genotype that maybe I have a high risk for developing some disease, I'm predisposed to it, but that doesn't mean that I'm going to develop it or I develop it at a certain age. It means that I have a risk for it, and that if I do things that are conducive to my sort of protection, then that means that I have a lower chance of getting that disease. And it depends on the effect of the genes that are involved. But, if I have a risk factor, say for like heart disease, and I go out and I eat a lot of steak, every week, it might increase my chances of actually getting heart disease. If I eat a very heart healthy diet, then I will probably have a lot lower risk of actually developing heart disease, even though my genes, I have a genotype that's predisposed to it. So, and these all relate back to the brain because we talk about phenotypes and we're talking about ultimately genes and their expression and the variation that you find in genes. And so those are things to keep in mind. You often run across genes in behavior studies, and so that's something useful. Also, we use model organisms to look at aging, and we wouldn't think that model organisms would be useful, but C. elegans is actually, and this is the nematode I was telling you about. It's a very small worm, and it's a very common model organism used in neuroscience and in biology. This is used as a model organism for aging, and it's interesting because they don't really live that long. They live a couple days, but what we find in C. elegans is a molecular model for understanding how aging works. And so the advantage using something like C. elegans or any model organism we usually use, we try to use a model organism that has advantages. It's either really close to humans in terms of its physiology, fairly close to humans, or it has a number of traits that allow you to manipulate the organism in ways that are easy to understand. So C. elegans falls into that latter category. And the thing about C. elegans is that we know what every cell does, and we know the number of cells are fixed in the adult. So we know the number of cells that will result in an adult worm. We know what they do. It has a fairly short lifespan, so we can study like interventions and look at the lifespan and understand like what the interventions do. In humans, you'd have to sit around for decades. But in C. elegans, you can do it in a couple days. And finally, and we talked about single cell sequencing last week, the sort of the genetics of C. elegans is highly tractable, meaning we know a lot about like what the mutants look like. So if you knock a gene out, we know what the phenotype looks like. We have a good handle on the genes and the function of the genes. And so it's all very good for understanding these things. And so, and plus C. elegans has a nice, there's a nice effect of lifespan. So we have wild type animals, which are basically the genotype that you have when you're born. And then they have these mutants where they actually take out genes, they remove genes, and they have these mutations that are defined in these strains that they have, they call mutant strains. And in some strains, you can get them to live much longer than the wild type. And so you have these mutants that are long lived. And you can study what the molecular pathways look like in those mutants. So that's C. elegans. That's a model organism, of course. But in general, aging is an actively regulated process. And so there are a couple of pathways that are sort of span from C. elegans to humans. And this is another reason why we use C. elegans, because even though they're very different animals, different organisms, they have similar molecular pathways. And so one of those pathways is IGF-1. And IGF-1 is it mediates metabolic and endocrine control. So it's not really anything that involves like neurons or neural signaling directly, but it's metabolic and endocrine control. And so those things will, affect metabolism, they'll affect inflammation, they'll affect basically how you respond to different stresses. So those are things that are that it doesn't look at first glance like that would be useful, but it actually is highly useful in understanding lifespan because it controls a lot of those functions that are not exactly at the cellular level, but just kind of sort of keeping those cells in good health. And so adaptations in this pathway have been observed to lengthen the lifespan. So I think I don't know if this is in this paper here, but they looked at centenarians where people who live to be 100, and they found that adaptations in this pathway can lengthen the lifespan. Another pathway is mTOR, which is this actually regulates cellular senescence. So we talked about cellular senescence before. mTOR pathway mediates mitochondrial maintenance, so that's the energetic balance in the organism, along with cellular growth, death, and proliferation. So mTOR actually has a stronger connection to neurons and like their basic overall health. But again, those things can affect the lifespan across the body. And so those are two pathways that people look at when they think about aging. And so it may seem a little disconnected from the brain, but it's definitely has a role in maintaining the physiology. Finally, there's this thing called life history theory. And so one of the things we find is that they're trade-offs. And so when I talk about like things like stress or inflammation, I'm really talking about trade-offs. And so in you and in me, our bodies are, we optimize how we distribute our energy in this physiology. And so life history theory suggests that if we don't get a lot of energy in our youth, or if we have things that happen in our youth, like a lot of chronic stress or chronic fatigue, things like that, it can affect us later in life. And the way it affects us later in life is how our physiology allocates resources, energetic resources to be specific. So you have this case where you have resources going into this juvenile, and then there's an allocation to survival and an allocation of fecundity. So in this case, this organism is resource rich. It doesn't really have to make this trade-off too much. It can survive and it can have a lot of offspring. But if there's a resource scarcity, then it's going to allocate a lot of its energy to survival and not so much to reproduction. And so this works also for physiology more directly, where you have this, if you're allocating things to survival, then what are you allocating it to? You might allocate, some of your energy to your immune system or to like physical growth. So bone, cartilage, and muscle. So imagine if you have like this organism and it's stressed all the time. Is it going to allocate a lot of its energy to the brain? Or is it going to allocate a lot of its energy to the rest of the body? Or is it going to allocate a lot of its energy to the immune system? That's the idea here. And so there's a trade-off, and this kind of drives aging, because if you don't have a lot of resources, or if they're allocated in one direction or another, it can lead to early neurodegeneration. And so finally, here's a graph showing this a little bit more concretely. This is where you have chronic disease, and this so this is a little bit difficult to see, but basically you have this. So this dotted line is a disease state, and so in the disease state versus the healthy, which is the solid line, you have a lower metabolic capacity in the disease state, and so this is early on, right around birth, and then you have a higher metabolic load in the juvenile stage, which is puberty. And so the combination of these two can lead to a reduction in height as an adult. So this is measuring height. So this is actually where you have lower metabolic capacity early, higher metabolic load later. This is just a constant stress on the organism, and it results in this reduction in height overall. So the energy is put into like sort of surviving this, and then you end up with this deficit later in life. And so you have this accelerated life history trajectory that disfavors later growth. The healthier trajectory, however, grows more slowly, and it incurs less metabolic load, but it favors growth later in life, or it gives you you end up being taller because the regulation here is you don't experience as much metabolic load. So I mean, this whole graph, I think like Anson in particular would appreciate this, because I think he talks a lot about like allostatic load and things like that, and I think this is very similar to that idea. So that the takeaway from this graph is that stresses early in life can result in changes in aging or changes in the rate of aging later in life. And so that's all I have on that presentation. So it was a little bit of a diversion from our typical computational stuff that we do, but I hope that was useful.
Will these slides be uploaded somewhere?
Yeah, I can send them out after the talk, or after the meeting.
I definitely want to go look at the last slide again, for the reason you mentioned.
Yeah. So I just wanted to give people like a little bit of neuroscience before the Neuromatch. I don't know how much I'll be talking about aging, but I think that's probably useful to understand a little bit about how, I think that's a good representation of like neuroscience, where you're not only talking about neurons, but you're talking about like genes and physiology and. Oh, Jesse, are you muted or?
Can you hear me now?
Yeah.
I have two different mutes. One on my headset and whatever else. I don't know what you heard, but I was saying, I think it's a good overview and it's a good way to tie in a bunch of stuff in neuroscience. I was curious, I had talked to Sunidhi before, but I don't know what specifically she was going to focus on. There's something else I was going to say too. I might say a few more things about Neuromatch after this, but yeah, okay. It's good.
Sebin looks like he's thinking about something.
Yeah, actually, I was just looking at what Jesse is saying. Actually, it was a really great presentation, and rather than the academic interest, it looked very scary for me, seeing the drop in our reasoning abilities and cognitive abilities over the age, and the impact of stress in the long time on our body and brain. Actually, it looked scary for me rather than my academic interest. It was a great presentation. Thank you.
Yeah, I'm like, I really need to get my sleep game in order, especially during Neuromatch, so I can not, be stressed and everything else, and some deep breaths and my Zen moments.
I just bought a heart rate monitor to do biofeedback on, so I'm gonna keep myself young. Yeah. Not too late for healthy, but young.
Well, yeah.
Okay.
So, were you gonna say something?
Bradley, I have a doubt. You showed, I missed the name of that you showed an organism with a small number of neurons as an example, like a case to study the effect of aging.
Yeah.
Can you show that slide if it's possible?
Yeah, I'll show it. So yeah, we actually work on this in my other group. This is the organism C. elegans.
Yeah, okay, the C elegans. Yeah, actually, where are you trying, actually, is it like that you are trying to prove that the C. elegans can be considered something in match with humans? Is it something like that you are trying to say? Or C. elegans is a completely different case?
Well, all right. Let's go back to that slide. So yeah, so we use model organisms actually in biology all the time, and the thing about C. elegans is that it's not. We don't really argue that it's exactly like humans, but so like if we use a rodent, the rodent has a lot of similarities with humans, like the brain. Like I showed back here, the rodent hippocampus versus the human hippocampus. The hippocampus, that's a common structure in both organisms. They look different, but they're common. C. elegans doesn't have that. They have a very different physiology. But what C. elegans does have is it does have some of the molecular pathways which are in common with humans. And also it's very, parallel to that, it's very easy to manipulate. So we can raise it in the lab, we can do like genetics, we can do all sorts of things. And so we use C. elegans actually. They use it for a number of different biomedical sort of parallels. I know they use it for aging. I can't remember some of the other ways that they use it, but I think aging is one of the primary models that they use C. elegans for. So it's yeah. I mean, if you study neuroscience, you encounter model organisms. So.
Your thoughts on the change in hippocampus among the taxi drivers was really insightful.
Yeah, it's like when you do something that's like spatial, hardcore spatial navigation. Like if you're always moving around in space, you develop the idea is you develop these really rich spatial representations. And so they do these studies where they like image the brain and then they look at the morphometry. They call it voxel morphometry, and they can see that like the size of it is increased relative to like a control, which would be like people who aren't taxi, maybe people just walking down the street who don't really have rich spatial representations. Now, I talked to someone about this once, and they didn't believe the study, so it's controversial somewhat. I mean, people fight about this all the time, but about different things in science. But it's worth thinking about. Like, if you have these really rich representations of things, like a spatial representation, does it mean that you need more anatomy in the correlation to like neural networks? Do you need a bigger neural network, or is it just enough just to have like a normal brain, and it's just more efficient at like processing or whatever? That's something that's an interesting thing you get out of that. Is like definitely there are a lot of studies on like expertise where people.
By what number will you be expecting a change among these drivers? I mean, about this spatial ability in the hippocampus from a common man, or I mean an average human. How much percentage of change or variance?
I don't think
it doesn't number. I have seen something.
Yeah, I don't think it's huge. I mean, I don't think they like doubles in size or whatever. But they'll do an analysis and they'll say, well, it's bigger, statistically significantly bigger, which is like, a little problematic sometimes because if you, you have your sample size is, not very big because, how many humans are there versus what's your sample size in the study. But yeah, with like expertise, we find that again and again that like with video, I talked about video games earlier, we can measure, I think, there have been a couple studies where they've measured enhanced spatial attention. So, like, if I am an expert at playing video games, you might present like something in the periphery of my vision, right? Like a laser light, and I have to recognize it. And so in people who don't play a lot of video games or don't have that heightened spatial attention, they don't recognize it way out on the periphery of their visual field. But like in people who play a lot of video games, they pick it up and they respond to it more quickly. And so that's interpreted as like this enhancement of their ability to like see a wider range of things out in front of them. Now that doesn't necessarily mean that there's more anatomy. It just means that they're using their resources more efficiently.
Thank you.
That's a really interesting general topic for me, because even thinking about my ongoing cat example, where the sort of stray cat is being domesticated in the house,
Yeah.
I've been thinking a bit about this in the sense of, in other random Jesse stories, in part of my spare time recently, I don't play video games much. I used to more when I was a kid, but I still, every now and then, but I'll watch people play them. So I actually was watching like professional video gaming tournaments, and it was a fighting game, and it was just interesting to see how, someone immediately after the game, and they're still in the zone of the game where they have very sort of filtered perception and filtered activity, and it sort of like made me think about the cat in the sense of the cat is changing its filters. So in the sense of, the video game analogy that you use is like, is someone expanding something, or are they like tweaking the filters to have more emphasis on certain parts of things? And like, there's a lot of tie-ins to direct perception and other stuff here.
Yeah.
So I'll just leave it at that. But this is kind of something that I've been thinking about. We'll come back to later.
Okay. Okay. Well, we're at the end of our meeting, I think. We're 20 minutes over the hour. But Bobby, did you have anything you wanted to add, or?
Maybe the account that he's not on right now. I think he's on.
Okay, that's okay. All right, guys. See you next week. Oh, actually, I'll give you some information about next week, because we have that presentation on Saturday. So maybe it'll be Sunday or something like that. I'll find another time, and then we'll see where we are with Neuromatch as well.
Yeah. I think in Slack I'll say things. I might update the Neuromatch Academy project board. There's a couple things there that I mean, anything like essential. It's just sort of better housekeeping stuff as I'm trying to keep track of all the stuff. And then the Notion thing, which I think I have a pretty good. What I showed you in Slack, or I think everybody in Slack, is like a version of like an existing database that I have, but I think there'd be a pretty easy way to do an accessible one for the lab to keep track of stuff. So that's good. But yeah, is it? I know Bobby came back. Is there anything that he wanted to say before we go?
Oh no, my internet just cut out for a second.
Okay.
Okay.
All right. Well, thanks for meeting, and hope you have a successful beginning to Neuromatch.
Yes.
Yeah.
Yeah, thank you.
Yeah. And we'll talk next week. We'll talk on Slack in the meantime.
Okay.
All right.
See you, everybody.
See ya. Bye.
All right, thank you.